Our next speaker is Shuang Li, hopefully I pronounced that correctly, and mechanism of non-genetic heterogeneity in yeast growth rate and stress resistance.
Okay, so hey, I'm Shuang from New York University, and today my topic will be mechanism of non-genetic heterogeneity in yeast growth rate and stress resistance. Good. So first, what is non-genetic heterogeneity? Basically, it means the variation in same trait exhibited by isogenic cells raised in a common and stable condition, and is of great clinical importance since here I'm showing you a picture of melanoma cells, and in this population there is actually a subpopulation of slow cycling cells showing by the arrows. And the slow cycling subpopulation is caused by increased expression level of a histone demethylase, so it's non-genetic. And in this paper, it proves that the slow cycling subpopulation is indispensable for continuous tumor growth, which means if you suppress this phenotype, the tumor will continue growth for days, but the growth will cannot sustain over weeks. Okay, so non-genetic heterogeneity is an interesting and important question or phenomenon. I'm going to study this in yeast with the aid of our high-throughput microscopy. Basically, what a microscope does is to track single cell growth and dividing over time. And in the meantime, you can calculate the growth rate for each one by the log of the area change over time. And in the best case scenario, basically you can track 5 times 10 to the 5th cell per plate. It means like simultaneously at the same time. Okay, so if I have a single cell growth rate calculated for each individual within the population or clonal population, what I can do next is to plot a histogram for the growth rate distribution. On the x-axis will be growth rate, and the y-axis will be the cell count. And here I'm showing you the data from the haploid AFY4 strain. That's basically the wild-type strain. And we can see that there is non-genetic heterogeneity showing here. Basically, here is a slow-growing subpopulation showing by the elevation of the left tail if you compare to the right one. So what are those cells? When I sorted for the cells, they can actually recover, for the slow-growing cells, they can actually recover the original growth rate within 48 hours. That means the slow-growing phenotype is not genetic. They can switch to the fast-growing genotype within several generations. Also, on top of that, I take a look at the petite cells. So in yeast, petite cells are spontaneously generating mutations, which is not of interest in this study. And unfortunately, the petite cell does fall in within the range of this left tail. So for the later plots, whenever relevant, I've already applied a mitochondrial stain to exclude petite from the further analysis to get more accurate data. Okay, just one more thing. So for the further plot, what I'm going to plot for the growth rate distribution is actually this cumulative density curve instead of the histogram for easier comparison. So how does it work? Basically, if the mean of the growth rate shifts, we'll see the shift of the curve. And if the shape of the growth rate distribution changes, like here, if we see a reduction of the slow-growing subpopulation, we'll see a reduce of the tail part of this curve. Okay, that's what we'll expect if the gene is only regulating the heterogeneity instead of the mean. Okay, so right now we know there's this non-genetic heterogeneity, and a previous postdoc, Sasha Levy, in our lab proves that the slow-growing cells actually bear a great biological importance. That means they can survive much better upon acute heat shock compared to the fast-growing ones. And also the slow-growing cells has an elevated expression level of TSL1, which is a subunit of a trait halosynthesis. So here I'm going to show you a movie clip to prove to confirm what I just told you. On the left-hand panel is a bright field channel, which shows you how the cells develop before and after heat shock. And on the right-hand panel will be the fluorescent channel, shows you the TSL1 expression level. Spoiler alert. So what you're going to see here is all these cells are going to be faster dividing, and this one here will be the slow-growing cells, and that will be the only one that survives after heat shock. Okay, let's see the movie first time. We can see everything's fast growing compared to this one, and that's only One survives after the heat shock, and it continues to divide for 15 hours afterwards. Then I'm going to play it again, and then we can focus on the right panel to see how the TSL1 level corresponds to the cells. And what are you going to see? Another spoiler alert. What you're going to see is the slow-growing cells will have a really high fluorescent intensity. It means high TSL1 expression level. Let's see it again. Here you can see it's really bright. And then after heat shock, that's only survivors. Okay, so hopefully I convince you that's what's happening. And if we put everything to a genetic network to make it more simpler, before such a study, what we know is that under stress condition, TSL1's expression level will be elevated, which will induce a stress response and which will cause the inhibition of cell growth. In such a study, what we know is that even under benign condition, we will see a heterogeneity of TSL1 expression level, and also we will see a heterogeneity in cell growth. And those two are negatively correlated, but TSL1 is not necessary for the slow-growing phenotype. Okay, so if we want to know how all this works, how all this is regulated, we'd better find candidates in the upstream regulation network. Before this study, what is known as MSN2 and MSN4 are transcription factors of the general stress response, and its function is regulated by intracellular cyclic AMP level through protein kinase A. And the cyclic AMP also have all these upstream regulators. So for the rest of the talk, I'm going to focus on these three aspects. Let's begin with the growth rate heterogeneity first. And I'm going to test firstly with MSN2 and MSN4 here. So here comes again the cumulative density curve. For a better visual effect, I'm going to transform the y-axis to a square root scale so that we can zoom in properly on the tail part and show you more details. And here, the red line I'm showing you is the wild type data. Basically, that's haploid A F54 strain. And then if I knock out MSN2 in this strain, what we're going to see is there's a significant reduction of the initial tail part here, which suggests there is a significant reduction of the slow-growing cell proportions within the population, and that will be exactly what we would expect if the gene is regulating heterogeneity instead of the mean. And if I knock out MSN4, what we see is a similar significant reduction, and if I knock out both, the reduction level is also similar to either of the single mutation. This suggests to me that MSN2 and MSN4 are each necessary in regulating the heterogeneity of the cell growth. Okay, then I want to take a step upstream to test the role of cyclic AMP. What I'm going to do is I'm going to knock out either IRA2 or PDE2 in the cell. And despite all the other pleiotropics, the common phenotype IRA2 mutant and the PDE2 mutant has is they both have elevated intracellular cyclic AMP level, but the degree of elevation may be different. Okay, so here I'm showing you the data. The orange line here shows the PDE2 mutant cumulative density curve, and the green line is RA2 mutant. So if we just ignore the shape of the mean and focus on the shape, we will see that both the PDE2 and RA2 cause even more significant reduction of the tail part compared to MSN2 and MSN4. This suggests to me that cyclic AMP definitely has a role in regulating the cell growth heterogeneity, but probably it's not solely through MSN2 and MSN4. Okay, so let's move to the second part of the heterogeneity of stress resistance. First, I'm going to focus on to see what are the regulators of TSL1 expression heterogeneity. On the x-axis, the growth rate, and y-axis, the per pixel fluorescent intensity for each cell, which is shown as a dot here. And I'm plotting for a non-fluorescent strain as a control, or the basal level. Here's the data for TSL1 GFP strain. We can see there's a significant negative correlation, which confirms our previous results. If I knock out MSN2 within this background, we can see that TSL1 expression level is bringing back to the basal level, and the negative correlation is abolished. But if you knock out MSN4, actually the strains still have a similar expression level of TSL1, and the negative correlation is preserved. Okay, so this suggests to me that only MSN2 but not MSN4 is responsible for regulating TSL1 heterogeneity under benign condition. Okay, then I want to test how are those mutants respond to acute heat shock. and whether it correlates to the TSL1 heterogeneity. And indeed, only MSN2 shows a significant reduction of the survival rate upon acute shock, but not MSN4. So this definitely, it does correlate with the TSL1 heterogeneous expression pattern. Then let's move to the last topic of the what's the molecular mechanism of the regulation. And I'm going to take MSN2 as a candidate and to study its effects on the heterogeneity of the cell growth. So there's the list of factors of the MSN2 we know right now. First is MSN2 is constitutively expressed. So the hypothesis, corresponding hypothesis will be probably MSN2 expression level correlates to single cell growth rate. The second fact we know is MSN2 shuttles in and out of the nucleus under stress. So the corresponding hypothesis will be probably MSN2 also shuttles under benign condition, and probably that's intracellular dynamics is what matters. Okay, let's take a look at the expression level first. So on the axis, I'm again plotting the growth rate. The y-axis is the per pixel fluorescence intensity, and again I'm plotting non-fluorescent strain for control. And here I'm showing you the data for MSN2 cherry strain. We can see that the average signal level does increase, but there's no significant level correlation between the growth rate and the MSN2 expression level. Okay, so it tells me expression level probably doesn't matter. Then let's take a look at the intracellular dynamics. So classic theory thinks MSN2 mostly stays in the cytoplasm under benign condition. But if you take a closer look here, I'm going to show you a clip of movie of MSN2 Amcheri strain under benign condition, and we can see it does shuttle, but it's under on a milder scale. We can see the MSN2 signal come together, form a focus that's in the nucleus, and it comes out and disperses in the cytoplasm. What I'm going to do is I'm going to track the same cells for MSN2 shuttling, and then I'm going to track exact the same cells longer for the growth rate calculation. Okay, here I'm going to show you first the tracking data. On x-axis the time scale, on the y-axis the fluorescent intensity of nucleus over cytoplasm. Basically, that's the index of the MSN2 nuclear occupancy. And the red line shows the track of one single cell over time. Just to show you, the track is faithfully reporting what's really happening. I'm going to show the cell, like real cell image for the highlighted dots. We can see here, like for time point 2 and time point 17, when the signal reported is low, MSN2 signal is like very dispersed in the cytoplasm. But for time point 8, which corresponds to the maximum like 1 point in the track, MSN2, most of MSN2 goes into the nucleus and form a bright focus. That tells you the track is working. And then what I'm going to do later is after the tracking, I'm going to track the same cell to calculate for longer and to calculate a growth rate. So if I'm plotting a growth rate on the axis and against MSN2 nuclear occupancy on the y-axis, we can see that there is a significant negative correlation between those two, which tells me that the slow-growing cells, in the slow-growing cells, there are more MSN2 goes to the nucleus and it tends to stay there longer. Okay, so if I sum it up, firstly, for the heterogeneity of cell growth, I proved that MSN2 and MSN4 are each necessary in the regulation, and also cyclic AMP also placed in a row here. And for TSL1 heterogeneity expression pattern under benign condition, only MSN2 but not MSN4 is required. And then for the molecular mechanism in regulating heterogeneity of cell growth, probably MSN2's intracellular dynamic is what matters. And with that, I'm going to give my thanks to all the Siegel Lab members, for the previous members and current members, and Pei-Ling for helping with the facts sorting, and Megan McLean for helping me with the initial construct for the MSN2 cherry strain. Thanks to all the funding sources, and thanks to the Yeast Genetic Group and to the organizers of TAGC to give me an opportunity here. With that, I'm going to take questions.
Well, we have time for one question.
Hi, that was fascinating. Have you looked at MSN2 and TSL1 in the same cells? Because the correlation wasn't super strong with MSN2. So do you think the TSL1 high expressors are a fraction or a subset of the cells that have high MSN2 in the nucleus, such that there's probably a second event that's needed to get high TSL1 expression, both MSN2 and then a subsequent event?
So unfortunately, I haven't really looked at MSN2's dynamics with the TSL1 like on a single cell level at the same time. But what I did is I actually sorted for enrichment of the slow-growing cells based on the TSL1 level. So can I go back? No. Oh, is there any way I can go back? Sorry. More. I cannot do it here, sorry. So it's just like the plot where I'm showing the negative correlation. That's where I do the sorting and enrich for the slow-growing cells with a TSL1 expression level. So after the sorting, we do see that those cells with high TSL1 expression level, they do have more MSN2 shuttling behavior. Is that what's your question?
Okay, yeah, but presumably in your cells that have low TSL expression, you'll also see some cells have high MSN2, which would fit the, right?
Yes, I think everything is on the probability scale. It's not absolute. There's no absolute because they absolutely MSN2 goes to this and it will cause that, but it's more on the probability scale.
Okay, thanks.
All right, thank you very much.